And now I would like to introduce our first speaker. Caitlyn Cruz is a master's student in the School of Environmental and Forest Sciences at the University of Washington. She works in the field of biofuels, transportation fuels, made from renewable resources. Her research examines what sorts of policies create the most beneficial conditions for biofuel facilities across the U.S., hoping to make the implementation of these fuels more widespread. Please join me in welcoming Caitlyn Cruz.
All right. Okay. Hi, everyone. All right. So we're going to start off with a question. So first, go ahead and raise your hand if you've driven a car today. Right. Now, how about if you've driven a car in the past week? Perfect. Now, go ahead and raise your hand if you've taken public transportation like a bus or a train in the past day or in the past week. Right. Now, here in Seattle, we're really lucky to have access to such a great public transportation system. But as much as I try to take the bus or the train, it can sometimes be just a little bit more convenient to drive instead. Now, you might have heard that taking public transportation like a bus or a train is better for the environment because there's fewer vehicles on the road, so emissions are decreased. And while this is true, it doesn't quite give us the whole story. Here in Washington, the transportation sector accounts for almost 45% of all of the state's greenhouse gas emissions. Now, this transportation sector includes both cars and public transportation. And it makes sense that if we want to reduce climate change and lower greenhouse gas emissions, to start with that largest contributor, transportation. So what can we do to improve this? Well, most of the fuels that we use in our vehicles today are fossil fuels. We have to dig these out from deep in the earth, like you can see in this picture, which can be hugely damaging to the environment. In addition to that, the stuff that we use to make fossil fuels is finite, meaning that we're going to run out of it at some point. Worst of all, fossil fuels have huge levels of carbon emissions. And the reason this is so bad can be summed up like this. You know how at night when it's cold, you just wrap yourself up in your coziest blanket? And you get all nice and cozy, and then in the middle of the night when we get too hot, we can just throw off the blanket, right? Well, the problem with fossil fuels is they act as a blanket over the atmosphere that we can't take off. Thus, they trap all the heat inside of this blanket, keeping the earth warming up with no heat able to escape. So that's the danger of fossil fuels. So what can we do to improve that? Well, luckily, we have technologies to make those same transportation fuels like gasoline or diesel, but from renewable materials instead. These fuels are called biofuels, and they're just made from renewable biomass like trees or plants. Now, the idea of using biomass for energy isn't a new concept. Have you ever built a campfire? If you have, you've used biomass as a form of energy. When these wood logs burn, they release energy in the form of heat. Now, biofuels have a lot of benefits when compared to fossil fuels, so we'll just run through some. Like I said, fossil fuels are dug out from deep within the earth, like you can see in this picture. This can be really, really damaging to the environment. But biofuels, on the other hand, since they're made from plants or trees, when they're harvested, they just come from the surface, making them a lot better for the environment. And again, since they're made from plants or trees, plants and trees can just grow right back, whereas fossil fuels, they're non-renewable. Most importantly, biofuels have really lower levels of carbon emissions than these fossil fuels do. Now, the level of reduction for carbon emissions actually depends on the type of fuel being used and on how those emissions are measured throughout the process of making that fuel. So to explain this, we'll do a little exercise. Go ahead and take a second. I know it's dinner time, but take a second and think about your ingredients that go on your most favorite sandwich. Make a mental list in your head. All right, got your ingredients? Now, your sandwich ingredients might include lettuce, tomato, lunch meats, or cheese. We can think of the ingredients that go on the inside of our sandwich like the emissions that are given off when a biofuel is being used to drive an actual car. but I have a friend who loves to make her own bread, so when she thinks of her favorite sandwich, she not only thinks of the ingredients that go inside the sandwich, but also the ingredients that go inside baking her bread, like eggs or butter. I'm not quite sure, I never quite got into that quarantine baking trend, but we can think of this additional step of the ingredients inside the bread as the emissions that are created during the production stage of making these biofuels. For me, my favorite sandwich is really easy. It's called the What's Up Doc, and it's the only veggie sub on a sandwich shop in South Carolina. When I was an undergrad, I ate this sandwich at least once a week. I wish that I was kidding. But just as important as that sandwich was, to make the meal really perfect, I needed a side of chips and a nice glass of cold sweet tea. For me, those fairly external ingredients were just as important as the actual sandwich. And we can think about emissions like that too. We can think of these fairly external sources of emissions, like how much emissions are being produced when biomass is being harvested or when it's being transported. Just like we all have pretty different ideas about what makes a perfect sandwich, producers of biofuels also have pretty different ideas about how much effort should be put in to reducing emissions at each stage of the biofuel production process. Now, depending on how much effort the producers put in at each stage, it makes the resulting fuels more or less a reduction of the carbon dioxide emissions when compared to fossil fuels. Now, this isn't to cause hesitation about biofuels. At the very least, most biofuels are carbon neutral, which means that they don't add any additional carbon into the atmosphere. But a lot of biofuels can actually be carbon negative, meaning that the process of making these fuels actually takes more carbon out of the atmosphere than it puts into it. The reason for this is because there are different types of biofuels. Now, most biofuels that we use today are called first-generation biofuels. These are made from agricultural or food crops, such as corn or soy. Now, I'm talking about all these benefits of biofuels, right? But there are some drawbacks to these first-generation biofuels. If we're taking up all the space to go ahead and plant all these crops for the purpose of biomass for biofuels, there is a chance that it could lead to possible deforestation, which isn't great for the earth. These crops also require a lot of water to grow, which can also strain our water resources. So how can we improve that? Well, I want you all to think about your drive over here to Town Hall. Did you pass any cornfields? What about trees? Here in Seattle, most of us probably passed a lot more trees than we did cornfields. Now, these first-generation food crops can be pretty finicky in terms of where they grow. The conditions have to be just right. But trees, those things will grow anywhere, which means that they're a lot more widely available. And luckily, we can make fuels out of them. These fuels are called second-generation biofuels. They're made from non-food crops like trees or agricultural waste. Now, these second-generation fuels are actually cheaper to produce and more efficient than those first-generation fuels, and they can be better for the environment. Now, most second-generation crops or second-generation biofuels are made from trees, but you can also use agricultural wastes. So, for example, if we harvest corn to eat, they take that whole corn plant out of the ground, break off the part that we eat, and then throw the rest and leave it in the field. But we can make fuels out of that. And that can be really helpful for using up that waste, but it can actually be better for the environment to make these second-generation biofuels out of trees instead. Let's go into why. So I want you to imagine that you own a company, right? And one day you find out that your company has accidentally dumped a ton of toxic chemicals into the ground. Whoops. So what can we do to fix this? Well, you could plant trees. As these trees grow, they suck up water from the ground like a straw. And in doing so, they can also suck up those toxins with it. As the trees suck up the toxins, they break them down into harmless components that they just store in the tree trunk or the leaves. And eventually, all of the toxins will be sucked out of the ground and the ground will be clean. Congratulations, you're not in trouble anymore. And what else? You can actually go ahead and sell these trees as biomass to produce biofuels, so you're making a larger profit. Since the trees you planted were new and weren't there before, they're taking additional carbon out of the atmosphere that wasn't being taken out before, meaning that the resulting fuels from these trees can be carbon negative. So most of the fuels that we use today are first-generation fossil fuels, but what can we do to try to transition more to these second-generation fossil fuels? Or biofuels, sorry. Well, when I came to the University of Washington, I joined a research team filled with brilliant engineers. They were all working on technologies and processes to create these second-generation biofuels from wood. Now, I'm not an engineer, and I wasn't quite sure of where I fit in on the team. I just knew that the work that they were doing was really cool. One day, I had a colleague give a presentation about a process that he had come up with to make making these fuels a little bit more efficient. I thought that his process was really cool, and I asked my advisor whether he thought that we could implement that process here in Washington State. He didn't have an answer for me. He wasn't sure if it was feasible or if there would be support. I spent the rest of that week pestering my research team trying to figure out an answer to this question, but no one knew the answer, and so I had figured out my place on the team. I wanted to understand how I could take the technologies and processes that my team was coming up with and then apply them into the real world. So my research looks at what policies are most helpful to the creation of biorefineries across the United States. Biorefineries are just facilities that produce these biofuels. By looking at other biorefineries across the country, I could kind of come up with a do's and don'ts list to understand what sorts of things are helpful or harmful when creating these facilities. Then I could take that do's and don'ts list and compare it to my team's work to see whether what we were doing was feasible here in Washington. So to start out my research, I looked at 4 separate refineries across the US, in Oregon, Iowa, Kansas, and Florida. I looked at the technical characteristics of these refineries, looking at what sorts of materials they were putting in, what kinds of fuels they were getting out, how many jobs these refineries created, things like that. I tried to keep as many of these technical characteristics as similar to my team's work as I could, so I could really identify what those crucial points were. By looking at these other refineries' characteristics, I could compare them to my team's characteristics, and if our characteristics looked to be better than theirs, then our refinery would likely be fairly feasible. The next step of my research was to conduct interviews with people across the biofuels industry. And by doing these interviews, I wanted to help contextualize those technical characteristics within a sort of policy context, figuring out what sorts of policies these people thought were most helpful, what sorts of groups tended to support it, what sorts of groups didn't. In my research, I found 3 major themes, so let's get into them. The first one is the importance of local community support. For biorefineries to be successful, I found that they needed local community support really early on in the process. And according to my interviews, most local communities are actually really willing to support these local biorefineries being built. And that's because a lot of these refineries bring really great economic benefits to the communities they come into. For example, most biorefineries aim to use local biomass, which means that for a local farmer or forest manager, you could be making additional profits that you weren't making before this refinery. Additionally, when this refinery is being built, the construction of it will bring temporary construction jobs, and then once it's created, there will be permanent refinery jobs. So, in general, local communities tend to be really supportive as long as the biorefineries work with them from really early on and make sure that their needs are met. Now, the next theme that kept coming up in my research was strong tax incentives. When I was researching policies across these states, I found 3 major types of policies that kept coming up. These types of policies were laws and regulations, tax exemptions and deductions, and tax credits. Now, it's really important that we understand the difference between these types of policies so we can understand why some can be more helpful than others. So to help explain this, I'm going to tell us a story. I have a younger brother. So cute, right? Now, when we were growing up, we were totally different students. I was the quiet choir nerd, and he was the super cool athlete. But he did struggle a bit with his grades. My parents tried everything they could to get his grades to go up. Originally, they tried out just telling him, "Kevin, you have to get good grades or else." That's how laws and regulations work. Governments tell these producers of fuels that they have to make biofuels or else. But we all know what it's like being told what to do. Not great, right? So laws and regulations can sometimes work, but a lot of times they're not the best option. Now, when simply telling my brother to get better grades wasn't working, my parents tried other strategies. Now, my brother always loved making and spending money. So they decided that whenever he got bad grades, he would have to pay them. So my brother could reduce the amount of money that he owed my parents by getting those better grades. That's how tax exemptions and deductions work. They work by reducing the amount of tax that a company owes if they make those cleaner fuels. Now, in addition to charging my brother for bad grades, my parents also paid him for his good grades. so he could increase the amount of money that he got to keep by getting better grades. That's how tax credits work. They can increase the amount of profit that a company can keep if they make those cleaner fuels. Now, for my brother, it worked, and he graduated high school, and he's actually about to graduate college in May with a degree in economics. I wonder why. Now, we can see, just like my brother, these tax credits and tax deductions work best when they're used together. Now, the final theme that came up in my research were conditions that made it cheaper to both produce and buy biofuels. Now, most biorefineries typically have these feedstocks or inputs, and they create biofuels from it, and that's it. But the thing is, as great as it is to help the environment, it sometimes isn't feasible if the actions we need to take to help the environment are more expensive or costly than what we do on our day-to-day. So we need to make it feasible, right? When I was researching these other refineries, I found that a lot of them were actually shutting down because they couldn't compete with the low price of fossil fuels. I was really worried about this, but my team reassured me that the work that we were doing was different. My team developed the concept of a Biohub, which is when instead of just producing biofuels from these feedstocks, a facility actually produces additional co-products, which are just products that are made alongside the fuels to be sold for a larger profit. So here you can see my team's base process. We start out with these inputs of poplar trees, reed canary grass, which is just an invasive grass species here in the Pacific Northwest, and hardwood residuals, which are just leftovers from timber production. These go into the processing, and out comes biofuels. And on top of the biofuels, my team has already created a way to make from these same inputs to create nanofibers. Now, nanofibers are just these small fibers that can be used in things like water filters. So already, my team has created an additional co-product to turn a higher profit. But in this processing, these wastes are created. So lignin on the bottom left is just this material that's held within cell walls, and during processing, it gets squeezed out and just goes to waste. Wood waste is just leftover wood pieces, and hydrogen is just hydrogen gas that's released into the air. Now, in the normal processing, these are all just wasted and thrown away or released, and nothing happens with them. But my team figured out ways to take these wastes and turn them into even more products to sell. So for the lignin waste on the bottom left, my team figured out that you can take it and burn it to create heat and energy. And the process of burning this lignin waste actually creates enough heat and energy to not only power the entire facility, but also additional energy that can be sold to the local electric grid. For the wood waste on the bottom right, my team figured out a way to take that and process it into xylitol, which is this chemical that can be used as an artificial sweetener in things like gum or peanut butter. And finally, my team figured out a way to trap that released hydrogen, process it, and turn it into formic acid, which is another type of chemical that can be used in things like antibacterial agents. Now, because they didn't have to purchase any additional materials to make those products, it's cutting down on the cost of producing these fuels. And because these additional products can be sold for extra money, since the company is making more of a profit, they can reduce the price of their biofuels, making them more easy to compete with the low price of those fossil fuels. So remember, I was working to figure out the feasibility of my team's project. So let's go through and see if my team checks these boxes. So first of all, local community support. Yes, my team's work meets this. My team is partnered with Lewis County, Washington, which is just a few hours south of Seattle. And they're really interested in the economic benefits that could come with a biorefinery being brought to their community. And what about the other two? Strong tax incentives and cheaper production and processing. My team's work also meets these. Washington just last year passed the state clean fuel standard, which is going to bring those tax incentives needed to really get the production of biofuels up and off the ground in the state of Washington. In addition to that, the state also has other policies that can help incentivize these biofuels being produced. And for the cheaper production and processing, because my team has come up with that process of figuring out how to take the waste of the original process and turn it into valuable co-products that can be sold alongside the fuels, that will make it cheaper for the team to produce the fuels and for consumers to buy these fuels. So overall, yes, all signs point to my team's work being feasible to be implemented here in the state of Washington, which is great news. But what does this mean overall? Now, I want you guys to think about when you're pumping your car, maybe you'll think back to this. Imagine that in a few years from now, you could be pumping your car, but instead of pumping gas, you could be pumping biofuels. Now, you could drive your same car or take the same bus or train, but you'd be having a much lower environmental impact. We can see that we have these technologies available to make these fuels, right? But we have to make sure that we don't get ahead of ourselves. We need to make sure that we also have the policies in place so that these fuels can really be successful on the market. Now, this lower environmental impact is what biofuels can do for us and why they're so important. Again, maybe one day you'll be pumping your fuel. Now, that's a great goal for us to have on the way to electrification, and that's why biofuels can be so important. Thank you.
Wonderful, thank you, Caitlyn. And our next speaker up is Saumya, who is a graduate student researcher in the University of Washington's Department of Lab Medicine and Pathology. Saumya is studying the immune response to an aggressive and often lethal skin cancer, Merkle cell carcinoma, in order to improve immune-based cancer treatments. Please join me in welcoming Saumya.
Okay, how many of you have ever had a pimple? Great, so you know what it's like. It shows up at the worst times. Morning of your college graduation, day of your prom, right when people are about to take lots of pictures of you. But you also know that it will go away in a few days, and you can have someone Photoshop all of those pictures and pretend it was never there. This, however, becomes a lot harder to do if this particular pimple didn't get the memo and keeps growing until you have a large red dome-shaped thing on your cheek. At this point, you would probably be rightfully concerned and have it checked out by your doctor. And while I hope that you hear something else, something less severe, you may hear that you have Merkle cell carcinoma, which is a very aggressive skin cancer. It kills about 30% of people who get it. So just to emphasize its aggressiveness a little bit more, melanoma, a skin cancer that you may be more familiar with, kills about 8% of people that get it. So you may be wondering, who gets this cancer? How worried should I be? Well, don't be too worried about every pimple that you get. About 1 in 130,000 people in the United States get it every year. So that's about the size of 2 football stadiums full of people. So at least even one person in this room getting it is very rare. And who gets it? Typically it's older white men. Why older? Because as we get older, our skin tends to break down and germs that can sometimes cause cancer get in. Also, as we get older, our immune system, which we'll come back to in a little bit, gets weaker and it doesn't fight the cancer cells that may develop as well. People with fairer skin typically have a higher chance of getting all skin cancer because skin cancers are caused by UV sunlight and any damage that it does to your skin. And why men more than women? We don't quite know that. That's just what we see. So 20% of Merkle cases are caused by UV sunlight, Well, the other 80%, the majority of cases, are caused by a virus, Merkle cell polyomavirus, which is very common, and we're all typically exposed to it as children and have living in our skin cells right now. So why don't we all get Merkle? To talk about that a little bit, let's talk about what viruses are and what viruses do. So this is a virus. It is a bunch of genes that are surrounded by protective proteins. These proteins allow the virus to travel through our body and not get broken down into pieces. It allows the virus to make it successfully into our cells and live there. So viruses want to get into our cells and make more copies of themselves. But where they need us is that they don't have the proteins that they need to actually make more copies of themselves. So they use the same machinery that is already in our cells and the way our cells make more copies of themselves. So Merkle cell polyomavirus causes cancer by coming into our genes. It sends the viral genes to the same place that our genes are. And most of the time it just makes more copies of itself, doesn't do a lot of damage, but sometimes a small part of the virus gets into our genes. And because all of this is happening in a very small place, it can cause a lot of disruptions. And the disruption that this causes sometimes is what leads our cells to make more copies of themselves uncontrollably. And that fundamentally is what cancer is. Just a bunch of cells that keep growing without any of the normal restrictions. So we don't unfortunately have a way of preventing this progression or from being infected with the virus in the first place, but we may have the next best thing, a way to determine what treatment is going to work best for each patient who gets this cancer. So before we can talk about predicting the best treatment, let's talk about how Merkle cell carcinoma is treated in the first place. So if it's caught early, we can just cut it out. Won't come back, no problem. If it unfortunately has grown a little bit too big or is somewhere that it's hard to cut out, we can treat it with radiation, which harms the genes in our cells enough that they can't function and they wither away. This makes the tumor smaller so that it's more easy to cut it out. If it has progressed past that state and broken off from the main tumor site and is in your blood traveling around, we can give patients chemotherapy, which are just drugs that kill any and all fast-growing cells, which is what cancer is. But we also have a lot of other cells in our body that grow very fast, like our skin cells, our intestinal cells, our hair cells, and that's why we hear that a lot of cancer patients have these side effects of losing their hair and having intestinal problems. So we don't want to use radiation and chemotherapy if we can avoid it. And in the past 5 years, we've had a new set of drugs called immunotherapy that have come on the market. And these drugs take advantage of our immune system to target the cancer specifically. So our immune system is a system that is designed to protect us from all kinds of harm. And we come across a lot of harm. We get a lot of germs, a lot of cancerous cells that may arise, and our immune system takes care of those without attacking our own cells. And so immunotherapies use this specificity to only attack the cancer without attacking our cells as well. So before we talk about immunotherapy, let's talk about the immune system a little bit more to make sure that we all understand it. So our immune system is our first line of defense. While the skin is not technically a part of your immune system, it is a physical barrier that can prevent germs from getting into your body. But like fences, we can have broken skin, wounds, things can get in through our skin. If things do get in, we have guard cells. that can patrol our blood and prevent any germs from actually getting into our cells. They just eliminate them as soon as they see them. But what about viruses that get into our cells anyway? Or cancer, which is just an abnormal version of our cells? We also have sniper cells, and sniper cells can see what is in our cells and then give signals to those cells to kill themselves. So they see any viruses that are in there, any infections, any cancerous bits, and they send signals that tell the cell that it's compromised and it needs to kill itself so that it doesn't damage the rest of the body. And so if our immune system doesn't see this cancer, that just means that the cancerous cell will continue to grow. So having a weak immune system increases your chance for getting this cancer. So you may be wondering, how do these sniper cells, or more formally called T cells, see virus-infected or cancerous cells? So just like we like to share our thoughts, feelings, skills with the world to find community, cancer cells, or all of our cells, also like to engage in a little show and tell and be friends with their neighbors. So all of our cells chew up a little bit of what's inside of them, and they show it on these proteins called receptors. So the little blue and purple things sticking out of the cell are the receptors, and the circles are the cell parts that it's showing. And our sniper cells, or T cells, recognize whatever is not supposed to be in our body through a complementary receptor. So we can think of these T cell receptors as keys, but they only fit locks that we don't have. So they fit the lock for the infected or cancerous cell, but not for the healthy cell. And that's how they attack what's not supposed to be there while sparing what is supposed to be there. And that is the basis of immunotherapy. So now that we know that, we can go back to the different types of immunotherapy. The first is that we can give drugs that can excite these cancer-specific T cells. And by exciting them, they allow them to send signals that tell the cancerous or the infected cell to kill itself. And this only occurs if we have enough of these T cells in the first place, because if we don't have these T cells, then no matter how much of the drug you give the patient, they're not going to be able to excite these cells because they're not there. So if we don't have enough of the cancer-specific or virus-specific T cells, we can either grow these cells in the lab and then give them to patients, or we can give them a lot of the virus bits or the cancerous bits through a vaccine, so that any T cells that they may have that have not seen the virus or the cancer yet can come out, be excited, and go fight the cancer. So how can we find these cancer-specific T cells to be able to know what immunotherapy to use? We use the same principles that the T cell uses. We take 4 of the components of the T cell receptor and the cancer and virus bits that it's showing and combine them together, and that's what we call a fake cell. We also add a fluorescent molecule onto this fake cell so that it stands out and is bright and shiny. And then we mix this fake cell with T cells that we've gotten from a patient's tumor or a patient's blood, and then these will bind to the fake cell. So we then have a machine that we can put in this cell formulation, and it will find the cells that are bound to the fake cell using the shiny fluorescent tag and separate them from all the cells that aren't bound. So all the cells that are cancer specific are going to be in one tube, all the cells that are not cancer specific will be in a different tube. So that's great, right? We can count cells And I wish that this is something that we could bring to all cancer patients and improve their chance of having their first treatment be the curative treatment. However, this is really hard to do. We can do it in the lab, but it requires a lot of expensive reagents, a lot of expensive machines to be able to do this. It requires a lot of people to spend days at a time just to count these T cells. All of this makes it really hard for us to bring this technology into the clinic. But that's ultimately what we want to do. We don't want our research to stay in the lab. We want it to make it to where it can help someone. So how can we do this? And this led me to think about what is unique about the T cells itself. How does the T cell recognize the cancer? How does it differentiate what is cancerous, what is not cancerous? The T cell receptor. Oops, wrong way. So the T cell receptors, as you can imagine, are very diverse since they have to recognize all of the different germs that we may encounter. So we can think of these T cell receptors as keys, as we have talked about before, and all the viruses and cancers, different things it needs to recognize as locks. So each lock can have multiple keys that fit it, right? Like our mailman doesn't go around with 1,000 different keys, but he has a universal key that can fit many different locks, and we all have our individual keys. However, despite this diversity, they can't be infinitely diverse because each of the keys that fits one lock still has to meet the same requirements to be able to catch and turn the lock itself. This is just like T cell receptors in that they can be very diverse and still recognize the same thing, but they can't be too diverse because they still need to identify the same components. Great, so how do we do this? How do we look at the T cell receptors? We can look at them using the fake cell, which is very hard, but we can also look at the instruction manual for the cells. And the instruction manual for the cells are the genes. They tell the cell what to do anytime it needs to perform a new task. The parts of the cell go back to the instruction manual, figure out what to make to be able to carry out that task. And each T cell receptor is also in the instruction manual, so each cell's manual is slightly different. And if we could read it, then we can know what T cell receptor each cell makes. So can we read it? Well, just unlike English, which has 26 letters, our cell's instruction manual we figured out has 4 letters, A, T, G, and C. And just like we can go to a dictionary and find out what a word means, a lot of scientists came together over 13 years to create a dictionary for the cell's instruction manual. And this effort was called the Human Genome Project, and its result is that we can take a string of about 100 letters and know what it means, know what it makes, know what the instructions tell you. And while I think a spelling bee champion perhaps could, I know that I couldn't take a 100-letter word, especially if it was just made up of 4 letters, and be able to tell what it means. But computers are great at this. They can take very repetitive things, find patterns, and tell us what they mean. We already use them for so much, from predicting the weather to finding a different route from your way home after work when there's an accident on the 5, to playing computer games, which I frustratingly always lose to the computer, which just tells you how good they are, not that I'm great at games. And they can also sort packages that are being mailed all over the world. So they can take in a lot of information that we give them, find similarities where we might not be able to see them, and sort them into different categories. So this is what I did in the lab. I took a patient's tumor sample, found all of the T cells that recognized Merkle cell carcinoma using the fake cells that we talked about. So at this point, I know what the T cells recognize, and I know what, and then I went further into the cell, got out the instruction manual that referred to the T cell receptor, the specific instructions for the T cell receptor, and then I gave those instructions to a computer, and I told it to find patterns. So the computer did this well. It put them into two groups. So here the circles refer to each T cell receptor, and the size of the circle is how many of those T cell receptors were in a particular patient. So the size gives us a quantification. And the lines between the circles means that the T cell receptors are very similar. So based on this, the computer is telling us that group 1 recognizes one part of the cancer-causing virus or the cancer itself, and group 2 recognizes something completely different. So then I went back and checked how well it did. So I color-coded all of the different receptors, since I knew where they came from because I'd used specific versions of the fake cells. And we saw that it did this very well. It organized T cells that recognized one part of the virus into one group, and T cells that recognized a different part of the virus in the second group. So what this tells me is that if I took an unknown T cell receptor instruction and gave it to this computer, where it places that in these piles will reflect the accurate specificity of the T cell. It will accurately tell me what the T cell recognizes. And we can use these patterns that it found to quantify how many virus-specific T cells there are in Merkle cell carcinoma patients. So what would this look like in clinic? We can take blood or tumor from a patient, get all of the instructions that make the T cell receptors without finding which ones are specific to Merkle cell carcinoma. And then we can overlay them onto these groups that we have already found. And we can count which ones overlap, and which ones overlap will tell us how many cancer-specific T cells a patient has. So when we ask this question, if they have enough cancer-specific T cells, we can give them the immunotherapy that activates the T cells and just helps their own cells fight the cancer better and control it. No extra things needed. If they don't have enough T cells, we have a few options. We can give them vaccines that will help their T cells be excited, bring out the rare ones. We could give them cancer-specific T cells directly. If that doesn't work, just grow them in the lab and inject them into the patient. Or we can talk about more conventional treatments like radiation and chemotherapy. And beyond this, developing this tool in itself also has a lot of different advantages. So the first part of building this predictor or this technology involves getting a lot of cancer-specific T cells. So in this process, if we see that patients with very specific T cells do very well, while patients who don't have those don't do well, then we can target those specifically and give the higher performing T cells to patients who don't have them. We also have a few different therapeutic vaccines going into clinical trials just in a couple weeks, actually. And this tool will also allow us to follow the patient's real time, to get their blood before they got the vaccine, get their blood after they get the vaccine, and see how much of their T cells are responding, how many new ones we have activated. So yeah, that I'd like to close by thanking all of my mentors who've helped me think through this proposal in my lab for helping with all of the experiments, and all of you for your attention. Thank you.
Wonderful. Thank you, Saumya. And our final speaker of the evening is going to be Miranda Mudge. Miranda is a graduate student and researcher in the Molecular and Cellular Biology Department at the University of Washington. She is studying proteins to better understand how bacteria influence and respond to their environment. Her research involves using proteins and bacteria as a tool for predicting harmful algal blooms. Please join me in welcoming Miranda.
Imagine it's a beautiful summer day in Washington and you decide to take advantage of the weather by gathering up all of your friends and family and heading out to the nearest lake or maybe even the ocean to enjoy a day on the water. As you're spending time with the people that you love, swimming, splashing around, maybe even boating, you notice that the water around you starts to change. What was once a clear, beautiful lake now starts to become thick with slimy green algae that gathers up around you. And over the course of the day, this becomes so thick that it gets to the point where you can't see your hand just below the surface of the water. What's happening is you're in the middle of what's called a harmful algal bloom. These occur worldwide, happening in both coastal waters and inland waters, such as lakes. And with the progression of climate change, they're becoming more frequent around the world and lasting longer and causing more damage. Harmful algal blooms occur when organisms called algae, which are normally so small that you can't see them without a microscope, grow rapidly to the point where you can now see them from space. As you can see, harmful algal blooms are literally a big problem. And as the harmful in the name suggests, these algal blooms come with a variety of dangerous consequences that occur worldwide, but I'm going to highlight a few that happen just in the US. In 2015, there was a harmful algal bloom off the coast of California that damaged the Dungeness crab industry, causing up to $48 million of damage as these crabs could no longer be harvested. This also progresses into recreational industries, where coasts become so thick and matted with water that people can no longer go there for their vacations, and this threatens a lot of jobs and has a lot of economical consequences. Additionally, these blooms can contaminate water sources, and in places like Lake Erie, where they're reoccurring on a yearly basis, they can cause $3 to $4 million of damage per year due to water contamination. Blooms can also cause a lot of environmental damage, as they can kill a variety of animals ranging from fish to mammals to birds. And they can also threaten our pets. A lot of pet deaths occur due to contaminated waters. There are also local consequences as well in the razor clam and shellfish industries, which in the past few years have been really hit by the harmful algal blooms that occur in Washington. And this threatens hundreds of jobs and causes $1 million of damage. So as you can see, there are a lot of environmental and economic consequences to these algal blooms. This also extends into the sphere of public health, as these blooms can release toxins into the water that can damage humans if they come in contact with them. And this could cause consequences such as asthma and skin irritation if your skin comes in contact with the water, or if you ingest the toxins in the form of eating shellfish that weren't caught ahead of time. This can lead to even more dangerous consequences such as brain damage or even death in the worst case scenario. So as you can see, harmful algal blooms definitely earn the name harmful. And you may even be thinking that because there's so much damage that can be caused, we should think about finding a way of getting rid of the algae altogether to avoid this type of problem. With that in mind, the algae would like for you to know that they are also beneficial organisms. For the purposes of this talk, this green blob here in the middle will represent our algae, and algae are responsible for producing 50 to 80% of the world's oxygen, making them the forest of the ocean. They also are plant-like organisms that count as a food source for much marine life, ranging from small phytoplankton or zooplankton all the way up to mammals and birds. And because of this, they form a staple in the environment, so getting rid of them would be actually a very bad thing. It's also worth noting that not all blooms are harmful or produce toxins. Most of the time, the algae exists in the water without any consequences as this base for the marine food web. So what we want to understand is what causes this progression of the algae from normal dormant conditions into a harmful algal bloom. Blooms occur when algae start to grow out of control. They go from being a normal part of the environment to taking over and becoming the dominant organism. And this is a big problem. But this type of growth doesn't occur spontaneously or without reason. Blooms occur in response to changes in their environment. And there can be a variety of changes that can lead to the progression of a bloom. These range from human consequences, such as urban runoff from cities that exist close to large bodies of water, or additionally agricultural runoff, so the use of fertilizers and farmlands that can then be transported to the water when it rains. Additionally, even if we were to get rid of all of the human factors, these blooms can still occur from normal environmental conditions such as increased sunlight that causes warmer temperatures in the summer, or the nutrient influx of shorebird droppings or mammal droppings in the water. It's almost like toxic algal blooms are looking for a perfect storm of events, but they don't care if that storm is hail or rain or snow. A lot of different causes can lead to the formation of these blooms. And stepping back and looking at the big picture, this makes things really complicated because it's hard to pin down one factor, meaning these blooms are unpredictable. This in turn causes consequences that make it difficult for current monitoring efforts. So if we consider a scenario where you have a lake that often has the occurrence of harmful algal blooms, and we want to monitor that, one method is to send out scientists to collect samples of the water and look for the presence of toxins. But this can be pretty labor intensive. So if we consider the example of even sampling once every week, this is still a lot of work. And in the case of a harmful algal bloom occurring on a Wednesday, when sampling occurs on a Monday, we experience 5 full days where the toxins are being released into the water and the public could be damaged because of this. So what we really need are a way to predict these blooms before they happen. That's where my lab comes in. Despite millions of dollars of research efforts and decades of research, looking at the algae directly to try and pinpoint a cause for these blooms hasn't worked. So for my research, we've decided to take a step back and look at this from the big picture view that the algae don't live alone in the water. They're actually in a complex community filled with millions of bacteria. These bacterial neighbors, even though they're much smaller, are often interacting with the algae, and because they live in the same water, they're responding to the same changes in the environment that trigger an algal bloom to occur. And so what this looks like is a complex community where there are interactions occurring between the algae and the bacteria. And if we want to determine what the algae are going to do in order to form a bloom, we can look for the close interactors of the algae in the form of bacteria that are interacting on a daily basis. So such as the friends, the family, the yoga partners. By looking at the close interactors of the algae, we can begin to predict their behavior because they're going to behave similarly since they spend so much time together. So in order to do this, we want to look for the behavior of the bacteria as an indicator for algal blooms. But how do we look at the behavior of bacteria? We can do this by looking at proteins. Bacteria change their behavior by changing their proteins. And proteins are like tiny machines in the cell that carry out all the work. They allow the cells to perform all of their actions. So for example, if a bacterial cell wants to grow, it makes proteins to build an internal structure. If it wants to swim, it makes proteins to build and power a tail. And if it wants to eat, it makes proteins that allow it to digest the food around it. And by changing these proteins quickly, the bacteria can change its behavior. So if we want to study their behavior, we study their proteins. Now, in order to identify bacteria that are most closely interacting with the algae, we need to look for a behavior in the algae that we can find in the bacteria to link these two together. To do this, we go back to the basis that algae behavior is driven by sunlight. Algae are photosynthetic, which means they take energy in from the sun, and they use that to make sugars that they can use as food. And so this type of metabolism, or energy process, occurs on a predictable day-night schedule. Proteins that are related to gathering energy from the sun increase during the day when there's sunlight, and once the algae wants to process these foods and the sun goes away, these proteins decrease. So what we can see over time is that you get this predictable cycle of behavior that's occurring within the algae. And we can look for that within the proteins of the bacteria with the simple concept of pattern recognition. So by looking for a behavior in the bacteria that mimics the algae, we can solidify the relationship between the two. So let's do this with the example of our photosynthesis proteins, which we have at the top in green. If we gather up all of our bacterial candidates, we can look for proteins within them to see if they match up with the algae. And I'll go through a few examples here. Starting with the yellow bacteria, which does seem to possess a curve within its proteins, but it doesn't quite match the green algae. Same with the orange. There's a little bit of a curve there, but it doesn't quite match. We could also expect within the green and blue bacteria that there will be proteins that increase, proteins that decrease, but they don't quite match up with what we're looking for. Same with the purple that flatlines. But what we anticipate is that there will be bacteria that are behaving like the algae in a way that matches up with these patterns of proteins. And indeed, when we overlay the bacteria and the algae proteins, you can see that they line up. These two behaviors match. And even though they don't overlay perfectly, what's really interesting about this relationship is that the green behavior is driven by the fact that algae is getting their energy from the sun. But most bacteria don't get their energy from the sun. They're getting it from digesting food that's around them. So the only reason this type of pattern would come up is because the two are closely interacting. This happens because algae and bacteria are like roommates that eat together. When the algae gets its energy from the sun and it makes sugars, it also releases waste into the water, like an offering of tomatoes, that can then be used by the bacteria as a form of food. And the bacteria in turn process these ingredients and generate their own waste that is like a recycling of the tomatoes into a salsa that can then be reused by the algae. And because of this, we have this interconnected relationship that strengthens the idea that behaviors in the bacteria can be used to predict behaviors in the algae, such as when it's going to bloom. So what we have now are bacterial behaviors that are consistent on a normal day that we can look for in our study. But we're not here to talk about a normal day. We're here to talk about harmful algal blooms, which for the bacteria are actually like a very bad day. It would be as if you woke up in the morning and you were going about your day, you grab the newspaper, you get a cup of coffee, and you glance out your window to see a single robot shooting lasers at buildings stomping down the street. Now, your next actions are going to determine if you survive this latest apocalyptic event of the 21st century. But I'll keep it simple for you. I'll give you two choices. The first choice of what you can do is nothing. You go back to drinking your coffee and you hope that somebody else will take care of this problem as you go about your day. The second option is to do anything at all. Let's say you run for the hills, you pack a bag and you get out of there before there's a full-on invasion. Now, if you're like me, you're probably going to choose the second option. It seems like the safest bet. And with bacteria, it's the same way. Bacteria change their behavior before a bloom occurs. Blooms occur in response to a change in the environment, which is like activating a single robot invader. And this progresses into a growth that is like a harmful algal bloom. As bacteria are going about their day, they sense this change in the environment and respond by changing their behavior, like packing a bag or getting out of the city. And this change in behavior results in a loss of pattern of the normal behavior that we would expect. And notably, this occurs before we get to a harmful algal bloom. So now we can use this type of establishment of a behavior and loss of that behavior as a predictor for when the bloom is going to occur. And this brings us back to the idea of pattern recognition. In order to sense this behavior and the loss of it, we first have to be able to detect it. And pattern detection depends upon the number of measurements you take. So if we consider the pattern at the bottom, we go through and we sample 1 measurement per day. What we're going to observe is this. Now, if you were given this data and you tried to plot it, you would probably think, well, that's a straight line. That's not interesting at all. But really, this actually isn't reflective of the pattern of behavior that we're seeing. It's just a result of taking 1 measurement per day of a pattern that's reoccurring on a daily basis. We can change this if we increase our number of measurements, so let's say 2 measurements per day. Now our observed pattern better matches the actual pattern in the bacteria. But this is highly dependent upon when we take those measurements. So if we stick to taking 2 measurements per day, but they end up at the center of the curve, now the line that we get still doesn't match as well as the actual pattern, despite those points being taken. So we did some math in the lab and came up with the idea that we need 6 measurements per day in order to accurately observe the pattern that's occurring in these bacteria. So 24 hours in a day divided by 6 measurements per day means that we would need to collect samples every 4 hours. And for my project, we decided to do this for 23 days to catch a bloom. And if you're thinking, wow, that sounds like a lot of work, you're absolutely right. It definitely was. But thankfully, this was my view. For my fieldwork, we headed out as a team to Orcas Island and set up on a dock where, let's see, here we go. Sorry about that. We set up on a dock on Orcas Island and set up a way of collecting water and from about 100 meters offshore and drawing it up to the dock where we could process it through a series of filters that allowed us to capture specifically the bacteria. And in this tiny sailboat, we set up a temporary lab, which you can see here, where we had stations for collecting and preserving water samples, stations for filtering the bacteria, stations for looking at the algae under the microscope. And in addition to this, we also had a probe collecting real-time data of the water, so water quality measurements such as temperature, amount of light, amount of salt, the kinds of things that could inform us of a change in the environment that would trigger a bloom. And I'm very thankful that throughout all of this work, even though it was very labor intensive, I got to work with a really awesome team of women who made these days a lot of fun, both when it was beautiful and sunny weather in the afternoon and also cold, dark, and rainy at the 1 a.m. time points. And through all of this, I'm happy to say that we did manage to catch a bloom. So this image on the left shows a filter which is packed with the algae at the height of the bloom. We also have in the center a microscope image of the algae, that's Cattosaurus, which is known to be bloom forming. And you can see that the amount of algae in the water as captured by our real-time probe increases to the levels of a harmful algal bloom. So at this point, we now have over 1,000 samples that are ready to be analyzed, where we have gathered up the bacteria from the water, taken them back to the lab, and they're now ready to be processed so that I can look for patterns in the bacterial behavior that can serve as a tool for predicting these blooms. In a way, we're translating these complicated patterns into the type of sensors that could act as like a warning for a bloom. So we're taking the patterns and translating them into a yes/no question. When the patterns are consistent on a normal day, it's like these proteins are dormant sensors that are lying in wait. And when the change in the environment occurs that triggers a bloom, these warning proteins change as the behavior changes, and it's like warning lights lighting up to warn us that a bloom is coming. Ideally, the future of this project is to turn these proteins into a tool that can be used to predict harmful algal blooms. And there are a couple different ways we see this happening. One is the yes-no question of patterns of behavior that could be like a dip test, that current monitors could go out into their local samples of water, take a little bit of water, and test for the presence or absence of these proteins. And in a more ideal scenario, we would automate this process using technology that's being built at the University of Washington, such as the Sea Glider and these bottom two photographs, that could be programmed to constantly monitor the water for these proteins. And in this way, we would be able to predict the harmful algal blooms in real time before they occur. With that, I'd like to thank you for your time and say that with summer right around the corner, make sure that if you spend some time in the water that you're aware of signs for harmful algal blooms and that you stay safe and keep your friends and family safe. And hopefully in the future we'll have a tool for warning you that the blooms are coming before they happen. Thank you.
All right, wonderful. Thank you, everybody. And we have some questions from you all now. I feel like we're on Oprah or something. All right, and I'm Kevin. I'm the instructor for the Engage course this year. That's the UW program that puts on this program. And I just want to thank you all for coming and joining our event. So the first question we have here is for Caitlyn. And the question is, I've heard of energy being created from compost. Have you studied that at all? And if so, do you know how it works and if it would be useful for any household energy needs?
Is this on? Okay, great. I have not studied compost being used for energy, but in general, biofuels can be made from any sort of organic material. So since compost is organic material, it should be able to be used for biomass. Typically, one of the things that is used really often is actually leftover oil waste from restaurants. So I feel like compost could probably fit in that category, but the problem with things like that is just a matter of scale. So likely it could be used for some small scale energy operations, but maybe not on a large commercial scale.
Awesome, thanks, Caitlyn. And the next question here is for Miranda. And the question is, are these toxic blooms related at all to the lack of biodiversity in the water?
That is an interesting question. I would say that toxic algal blooms aren't limited to one type of algae. There's actually a lot of competition between the different types of algae, which is a reflector of biodiversity in the water. So I think that, give it some real thought here. I would say that I wouldn't limit that as a potential cause, but I think other environmental factors are probably a larger contributor to the formation of algal blooms.
Awesome, thanks, Miranda. The next question here is for Saumya. What led you to cancer research and particularly skin cancer?
So I think my path to cancer research or skin cancer research was very indirect. I was very interested in immunology. I first learned about TB, which hides, or tuberculosis, which hides in our cells and can evade the immune system, which is designed to recognize it and fight it. And with that, I was so fascinated by immunology that that's what I wanted to study when I entered graduate school. And I ended up in a skin cancer lab because they were studying a lot of immunotherapy, and because immunotherapy is becoming so important for cancer treatment in general. So I guess I got to studying cancer via immunology.
Awesome, thanks so much. And another question looks like for Caitlyn. Is the environmental impact really lessened if we're growing huge monocrops for biofuel?
Yeah, that's a really good question, and definitely a question that I also had when I started my biofuels research. And typically the answer is actually yes, and it depends on the way that you grow them. The goal for a lot of these second generation biofuels where we are using woody crops is to grow specific like new stands of trees. So we don't want to be disturbing any land that already has a good amount of forest growing on it. But like the example that I gave in my presentation, these tree lands could be used on sites that were already damaged, and so you could actually be repairing that site. And there's actually a lot of different tree crops that you could use to produce biofuels, so it wouldn't have to be a monocrop. You could plant a bunch of different types of trees and other plants as well on that one site to have that level of biodiversity.
Awesome, thanks, Caitlyn. Here's another one for Miranda. What is the protein in the bacteria that is changing and letting you know a bloom is coming?
That's a really good question. Right now, we're trying to identify which proteins are good candidates for this by looking for those that mimic the algae. But the list isn't just one or two. Ideally, we would like to have maybe up to 100 of these so that if one particular protein, for example, isn't present in the bacteria that are in the water, we would still have other candidates that we could look for. And additionally, we're not looking for specific bacteria, rather the proteins that reflect the actions that they're doing, so that if you do have a more limited community of bacteria, we could still look to the actions that are shared between the different types of bacteria to find a candidate. But we're probably thinking, or we are thinking, that these candidate proteins are going to be related to whatever change in the environment is causing the specific bloom. So for example, if there's an influx of nitrogen, we would see nitrogen processing proteins, something like that.
Awesome, thanks, Miranda. And then here's another one for Saumya. What cancer rates trending, are cancer rates trending up among the American population?
Yeah, they are. So most cancer rates are going up, but particularly for Merkle cell carcinoma, our population is aging. So as the years go by, more people in the US are older. We have a lower population of younger adults. So because this is an age-associated cancer, this is growing particularly fast.
Great, thank you. All right, and here's one for Caitlyn. This one's got a couple parts to it. And the question is, I'm curious about carbon negative fuels that you talked about. It says plants release carbon when harvested. And of course, there's greenhouse gas emissions when the fuels are burned. So in order to be carbon negative, they must sequester a huge amount of carbon before being harvested, right? What types of plants can absorb this much carbon in order to be carbon negative?
Yeah, that is a really good question. And I will preface this by saying I don't know a ton of them off the top of my head, but I know that in the lab that I'm working in, a lot of research is being done into poplar trees. And the way, especially to make sure that these fuels are carbon negative, is that if you're going to plant a bunch of different trees on a site, the goal would be to plant them in stages so that you're not ever going to be harvesting all of the trees off of the site at once. So you're always going to have some remaining trees still sequestering carbon. And the interesting thing about the way that the trees grow is that younger trees, because, like when we're growing up, we grow so much faster when we're younger, and then we grow a lot slower once we're older. So trees are the same way. They actually sequester a lot more carbon when they're younger trees. So by planting those new stands of trees, they do sequester huge amounts of carbon. And then by making sure we don't cut down all those trees at once, we can make sure that those resulting fuels from that patch are carbon negative.
Great, thank you. Here's another one coming Miranda's way. It says, if bacteria need to be measured around 6 times a day to determine patterns to predict algal blooms, are you suggesting that all bodies of water need to be tested 6 or more times a day on an ongoing basis? Or do you imagine being able to simplify or streamline the process at some point?
Yes, I wouldn't force that amount of work on anybody who's doing this type of monitoring. Ideally, we would look for a pattern that we could establish that would then go away. And so that would turn this into a yes-no type of question where we could develop technology to detect these proteins. And as soon as they're gone, we would know that a bloom is coming. But in a more ideal scenario, we would want to automate the process. And so that would be deploying a probe to these areas that could do constant monitoring so that humans don't have to do that.
Great, thank you. And it looks like we've got maybe one final question for Saumya. And it says, can your method of treating MCC also be used to treat other skin cancers or cancers in other parts of the body? What are the similarities or differences?
Sorry, can you repeat the first part of that?
Yeah, for sure. It just says, can your method of treating Merkle cell carcinoma also be used to treat other skin cancers or other cancers elsewhere in the body? And what are some similarities or differences?
Yeah, definitely. So immunotherapies in general are being used for lots of different cancers. They do particularly well for blood-based cancers. They're being used for melanoma, which is another skin cancer. And we're constantly expanding immunotherapies in general to other cancers in different parts of the body. And so the tool that I'm working on developing, we selected Merkle cell carcinoma partly because the virus that causes it is very small, and it allows us a lot of room to comprehensively study the immune system and the immune response to that particular cancer-causing virus. But there are also a lot of other viruses that are bigger that cause different cancers. So if this is something that we show to be feasible, we then have more rationale for doing all of the work to develop similar tools for other cancers that are caused by viruses, and to address other cancers that are not caused by viruses. A lot of people are doing studies to look at the T cells themselves, and look inside the cells to be able to say whether a T cell is cancer specific or not. So once we have those technologies available more readily, we can expand this T cell receptor based tool to those cancers as well. So hopefully this will serve as proof of concept and tell us that we should be doing this and should expand it to other harder to study cancers.
Awesome. Thank you to all three of you. And thanks for everybody here in person and online for submitting questions. And I think that's about all we've got for tonight. So if you liked what you saw, please join us for our other 4 events later on this month and next month. And let's give one final round of applause to our speakers for tonight.